and welcome to Spotlight on Antibodies, a podcast from BioIntron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in October, we'll highlight advances in antibody design with AI, crossing the blood-brain barrier, and universal antibodies. As always, links to sources are in the episode description. Artificial intelligence has officially entered the antibody discovery pipeline. Across the industry, AI is being used to predict and even design entirely new antibodies from scratch. Typically, antibody researchers use methods like phage display, hybridoma technology, and immunization to find leads. But now, AI can generate whole antibody sequences in silico. These next-generation platforms combine predictive modeling with wet lab validation. They can analyze massive datasets of antibody sequences, predict how a molecule might fold, propose beneficial mutations, and rank the best candidates for synthesis. The result? A much broader exploration of the antibody landscape. AI's evolution in this space mirrors the wider revolution in computational biology. Early tools used machine learning to predict protein folding or estimate binding affinity. But today's systems, built on transformers, diffusion models, and reinforcement learning, go far beyond that. One turning point came with DeepMind's AlphaFold, a platform with the ability to predict protein structures with near experimental accuracy. From there, generative AI models trained on huge antibody datasets began producing new designs. Some can start with an epitope, the region of the antigen you want to hit, and build an antibody backwards from it. That's the concept of de novo design, creating antibodies that have never existed in nature, but are shaped by data and optimized by performance. This isn't just theory, as AI-designed antibodies have now entered into clinical development. For example, imnescibart, also known as AU-007, from Alos Bioscience. It's the first fully computationally designed antibody to enter human trials. By selectively targeting the IL-2 receptor interface, imnescibart helps boost the immune system's cancer-fighting T cells while avoiding the usual side effects of IL-2 therapy. It's a powerful proof of concept for AI-enabled design. Recently, there have been several other news stories about this idea. Zyra Therapeutics launched with $1 billion in funding this year to advanced AI-driven biologics. Absci's integrated platform generates billions of protein variants and validates them thousands of times faster than conventional assays. Nabla Bio and Chai Discovery are also designing antibodies against targets once thought undruggable. The blood-brain barrier has long been a hurdle for antibody therapeutics in reaching the central nervous system. Its tight endothelial junctions maintain brain homeostasis, but severely limit entry of large biologics. This limitation historically discouraged antibody-based approaches for neurological disease. However, advances in antibody engineering and improved understanding of the central nervous system immunology now support the development of molecules capable of penetrating or bypassing the barrier. Studies in neurodegenerative disorders show that brain antigens can exit through lymphatic vessels and trigger peripheral immune responses, producing antibodies that can sometimes re-enter the CNS. A critical advancement is better modeling techniques. Traditional two-dimensional cell cultures and animal models rarely capture the complexity of the human blood-brain barrier. New three-dimensional in vitro systems like microfluidic chips, hydrogels, and brain organoids can offer more physiologically relevant tools for evaluating antibody permeability and receptor-mediated transport. Among delivery strategies, receptor-mediated transcytosis, or RMT, is leading progress by engaging receptors such as the transferrin receptor, insulin receptor, or FcRn. Engineered antibodies can utilize endogenous transport pathways to cross endothelial cells. Affinity and receptor selection are key. Transferrin receptor targeting improves uptake but can cause hematologic side effects, while FcRn engineering enhances CNS exposure, and extends half-life when optimized for neutral pH binding. Transport across the barrier is only part of the challenge. Once inside, antibody distribution within the brain parenchyma depends on diffusion, target internalization, and efflux. Computational modeling and molecular anchoring strategies are being applied to improve local retention and target engagement. Alternative approaches are also emerging. Peptide-mediated delivery using rabies virus glycoprotein sequences has enhanced blood-brain barrier permeability in models of viral encephalitis. And non-invasive intranasal administration has shown effective regional delivery of anti-IL-1 beta antibodies while reducing systemic exposure. Together, these advances illustrate a shift from viewing the blood-brain barrier as an absolute barrier to treating it as a biological interface that can be rationally navigated through molecular and delivery engineering. Antibody therapeutics have become essential for treating infectious and immune-mediated diseases, but most remain narrowly targeted, usually recognizing a single strain or antigen. Researchers are now working towards what are being called universal antibodies, molecules that act broadly across viral variants, parasite species, and even tumor types by focusing on conserved molecular targets. In HIV research, scientists reported the antibody 04A06, which neutralized almost 99% of over 300 HIV strains. Derived from elite neutralizers, this antibody reaches conserved regions of the HIV envelope protein that most antibodies cannot access, offering durable suppression and resistance to viral escape. For influenza, teams at the Jackson Laboratory demonstrated that antibodies targeting the conserved M2E region protect across human, avian, and swine strains. Instead of direct neutralization, these antibodies clear infection by engaging immune cells, showing that Fc-mediated functions can also deliver broad protection. In malaria, a first-in-human study tested MAM01, an antibody against a conserved epitope of the Plasmodium falciparum circumsporozoite protein. A single injection completely prevented infection after mosquito exposure, with protection lasting for months. The same concept is extending into oncology. Researchers created Unibody, which expresses antibody Fc fragments on tumor surfaces to attract immune attack, while an UC Irvine team developed GlyTR, a bispecific molecule that binds tumor-associated glycans and recruits T cells. Both aim to make immune targeting less dependent on any one antigen. Across these studies, a consistent trend is clear. Focusing on conserved biologic structures can yield antibodies with cross-strain, cross-species, and even cross-tumor activity. Universal antibodies may not replace precision therapeutics, but they could become a new class of broadly effective, escape-resistant biologics. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with future episodes from Spotlight on Antibodies by following this podcast.